Depression is a terrible problem to have, but research has shown that when used in combination with psychotherapy, meaning talk therapy, antidepressant drugs like Prozac are some of the most effective treatments for depression that researchers have discovered. So you might be asking yourself, how does that work? How is that even possible for a person to ingest a chemical and for it to totally change their mood and their outlook on the world? Well, like I said, usually it doesn't happen in isolation. For most people, the most potent, powerful effect comes when combining different treatments for depression or for other psychiatric issues like anxiety and other mood disorders. But it's still the case that Prozac and other antidepressants have a powerful effect. So how do they do that? What are they doing to your brain? Well, the answer comes down to a fundamental process in your brain called synaptic transmission. This is the process by which neurons actually communicate with each other. In a previous video, we talked about the action potential. And the action potential, also known as firing or spiking or turning on or activating a neuron, is when a neuron activates, it basically gets ready to send those chemicals. But the actual sending of those chemicals is the communication between neurons. And those chemicals are called neurotransmitters. Now, also in the last video, we talked about how when a neuron binds enough of a neurotransmitter, it can cause it to undergo an action potential and thereby sort of repeat the process, allowing that neuron to send its own neurotransmitters. Now, the details differ with regard to the various types of neurotransmitters about how all that works. But in today's video, we are going to talk about how that process happens, how synaptic transmission happens. By the way, if we haven't met, I'm Andrew and this is Sense of Mind. This channel is all about helping you to learn about your brain so you can upgrade your mind and improve your life. It's all about giving you fundamental knowledge about neuroscience as well as psychology and sort of other areas of the science of mind and brain, and allow you to incorporate that knowledge and then make better decisions about how to best live your life. So the video you're watching right now is part of a new series that will basically serve as an introduction to neuroscience. We'll be going from the very small, starting with the cells of the nervous system, mostly neurons, and then we'll be moving out. We'll be talking about circuits, we'll get into brain systems and brain regions, we'll be talking about cognitive neuroscience. So we'll really build out, and it's aimed at giving you kind of a fundamental knowledge of how the brain works, and so that you can move into more advanced topics on your own if you want. And this is the level 3 or sort of more advanced video in this topic. We also have 2 other videos. There's a 30-second version, which kind of just gives you a 30,000-foot view and a summary of the main points of what's important about synaptic transmission. And then we have a little bit longer video, but shorter than this one, that is the level 2 intermediate video, and that one goes into quite a bit more detail than the 30-second version, but not as much technicals as you'll find in this video. Now, to be clear, with all this stuff, you can go as deep as you want and far deeper than I go even in this more advanced video. So there are further readings and some other content that you can explore in the description of this video if you're not satisfied and want to understand more about neurochemistry and synaptic transmission and all of that. In the next video, our next set of videos, we will be talking about the different neurotransmitter systems. So we'll be talking about serotonin and dopamine, norepinephrine, GABA, glutamate. Those are the main ones, but we'll mention some others. And we'll talk about how these differ, and some of that will become clear later in this video, why we're going to talk about those. But for this video, we're just going to talk about that sort of biological process by which 2 neurons communicate chemically, and that is called synaptic transmission. So Without further ado, let's get into synaptic transmission. If you don't know what a neuron is or what an action potential is, check out these earlier videos we did on that topic. Again, there's 3 different levels of difficulty, so you can dive in at whatever level you want and learn about them. Really, any of them should be okay for this video, but understanding what a neuron is and what an action potential is pretty important for understanding this video. Okay, so in this level 3 advanced video, we will be talking about 2 aspects of synaptic transmission. One is called exocytosis and the other is enzymatic degradation. Later on, we'll return to antidepressant drugs like Prozac and other SSRIs, explain how they work, and it will make a lot more sense why taking a chemical like that can help some depressed people to feel better. Okay, so this is just kind of a graphical overview of the last video of the action potential. Again, this is super simplified, so if you want more detail, check out that level 3 video. But anyway, on the right, you see that neurotransmitter receptor receiving a neurotransmitter, and that causes changes in the concentration of ions, which is basically a voltage change in the neuron, and that can result in an action potential, which is that yellow kind of flashpoint thing right there. And that starts in the axon hillock, travels through the axon, eventually ends at the tips of the axon, and releases a neurotransmitter. So in that last video, we kind of ended here. And so on the right, we have the tip of the axon, and on the left, sort of the rest of the axon. And we showed that an increase in sodium ions caused a neurotransmitter package, we called it, to be released from the neuron. So what we want to know is how that process happens. How does the increase in sodium due to the action potential cause the release of the synaptic vesicle, the neurotransmitter package? All right, let's get into it. Okay, so once again, this is our neuron. We have on the right the axon, which is the cable that the neuron uses to send a message. We have the cell body, which is kind of the central area. There's a lot going on there. I have receiver written there because it can receive neurotransmitters. It has the right receptors for those. And dendrites. So there's a lot more dendrites than there are axons. And dendrites are also receivers of neurotransmitters. All right, so now what we're going to do is just zoom in on the interaction between 2 neurons. So you can see on the left we have our sender neuron. This is the one that's going to be sending the neurotransmitter. And then we have the neuron on the right, which is the receiver neuron. It is going to be receiving the neurotransmitter. And then there's this little tiny space between the 2 cells called the synapse. And this is kind of where the 2 neurons meet. And it really is a space. They're not actually touching, really. And so that little space is called the synapse. So the neuron that sends the neurotransmitter is called the presynaptic neuron. And the one that receives it is called the postsynaptic neuron, because presynaptic meaning before the synapse and postsynaptic meaning after the synapse. So it makes sense if you think about the direction of communication. The presynaptic neuron is sending the neurotransmitter across the synapse, then the postsynaptic neuron is receiving it, and that is why they're named as they are. Okay, so now we zoom in on that, right? We've got on the left our presynaptic neuron, and on the right the postsynaptic receiver neuron. And then that little space in the middle is the synapse. So where we left off was sodium was entering into the axon tip, or what we call the terminal bouton. And you see the sodium coming in, and what it's going to do is interact with these calcium ion channels. So these are basically tubes that are embedded into the cell membrane, and they cross from one side to the other, and they allow only calcium through. So and and they are also what is called voltage gated. So they're sensitive to changes in voltage. And when a certain voltage is reached, when it gets high enough, it causes them to open and allow calcium to flow in. So when sodium comes in, sodium is a positively charged ion, meaning it carries an electrical charge. So it raises the voltage in the axon tip, the terminal bouton, and that causes a reaction on the voltage gated calcium ion channels and opens them up and allows calcium to flow into the cell. Okay, so now calcium has flowed in, and what does calcium do? Why is it important? Well, when we talked about those synaptic vesicles before, those are the neurotransmitter packages. So they are the little bubbles or bags that contain the molecules that we call neurotransmitters that neurons use to communicate. And those little synaptic vesicles are actually held on these structures called microtubules. Microtubules are kind of what they sound like. They're these little tubes, little pieces of sort of cellular skeleton that can be used to transport materials across the cell and can be used for sort of storage. And in this case, they are holding onto these synaptic vesicles. So when calcium enters the cell, what it does is actually causes those synaptic vesicles to be released from the microtubules. So they pop off the microtubules, and now the synaptic vesicles start heading toward the axon tip, toward the terminal bouton. Okay. So now what has happened is all these synaptic vesicles are docking. This is a process called docking. They've lined up at the terminal bouton, and just to be clear, there would be many more synaptic vesicles, and they each contain many neurotransmitters, but we don't want to get too crazy. So this is what's going on. They've lined up at the end of the axon, at the terminal bouton. And so now let's zoom in to this area, this red square, and see what's going on here. Okay, so once again, we've got these lined up at the cell membrane. And now an important thing to note is that the bubbles, the little bubbles that the synaptic vesicles are made of, is actually the same material as the cell is made of. The cell membrane is made of. So they're basically like these little tiny mini cells inside the cell that are holding just neurotransmitters. So they're kind of a bag within a bag. And I think the best way to think about them is as like soap bubbles. So if you've ever seen a soap bubble, and if you've ever seen two of them come together and join into a bigger bubble, you know that what's going on is they're made of the same material, right? Soap. And they hit each other and they fuse, and then they just become one larger bubble. Now you can also imagine reversing that process, and so just keeping that in mind will let you understand what's going on here. Okay, so then what happens is those synaptic vesicles fuse to the cell membrane. So they do their docking thing, and then you can see a little space opens up. If you're looking at that top synaptic vesicle, basically what's going on there is it's fusing with the cell membrane, and that causes it to release its neurotransmitters into the synapse. So it has now released those neurotransmitters into the space between the neurons, the synapse. And all right, what is going on here? So we've got a bunch of neurotransmitters that have been released, and we have these two little orange things on the right, on the postsynaptic receiving neuron. So what are those? Those are receptors. So those are these protein complexes, basically, that catch specific neurotransmitters. And so these neurotransmitters float across, and a couple of them bind to that postsynaptic cell. And so just as we saw in the action potential video, this can cause a transient rise in the amount of sodium. So an influx of sodium, which corresponds to, as we talked about, a rise in voltage, because sodium ions, just like all ions, carry an electric charge. So bringing in positive ions will raise the voltage of that neuron just a little bit in that local area. If that neuron binds enough of that neurotransmitter, it will raise the voltage a lot, and that can trigger a full-on action potential. So then the cycle can repeat itself. So that is what we would call exocytosis. That is the process of a neuron releasing neurotransmitters and then them floating across the synapse and binding to the receptors on the other side. So one thing to keep in mind is there is a lot going on in just this little process. Just like everything in biology, the biochemistry and biophysics just gets deeper and deeper, and you can go into specifically which molecules and other ions that I didn't mention are involved in the process of exocytosis. There's a lot more to the synapse than just these two neurons, as we'll see in a second, but this is simplified, but this gives you an idea of how it works. So like I said, there'll be further reading and other content if you want to learn more about synaptic transmission and exocytosis specifically. But let's move on. Okay, so one other thing to keep in mind is, like I mentioned, so since the synaptic vesicles are made of the same material as the cell membrane, that allows them to fuse, right? That allows them to fuse because that material is the same, so it's just like two bubbles fusing together. But what happens there is that the cell membrane then has more material, right? Because you added to it, and so it can use that extra material for more synaptic vesicles. So there you see one just arriving into the cell, and it can be recycled for later use. Okay. Now, let's zoom in on the process of the neurotransmitter floating across the synapse and being bound by the postsynaptic receptors. Okay, so as we said, those two neurotransmitters bound to their receptors, but we're now left with extra neurotransmitter. It's just in that synaptic space, and we don't want to just leave it there. That's a waste, and it could lead to other problems like inflammation and other things. But let's see what can we do about that. All right, well, we need to get rid of it somehow. We want to clean it up and get it out of there. And the simplest mechanism that it's not that much more complicated than the other mechanisms, but it is one of the important mechanisms for getting rid of that extra neurotransmitter. And it's called autoreceptor reuptake, or just reuptake. And that is when a specialized receptor on the surface of the presynaptic neuron, the neuron that originally sent the neurotransmitter, it's a receptor that's specialized for basically grabbing those extra neurotransmitters and shuttling them into the cell. So you can see how it would start to suck up these extra neurotransmitters. And to be clear, in all these cases, you just got to multiply the number of receptors and neurotransmitters by, a gazillion. There's many more in there. I don't know exactly how many off the top of my head, but it's a lot more than 3. Anyway, so now we can take a quick break and talk about how antidepressants work. Okay, so it's pretty simple, actually. If we think about what's going on in this system, let's imagine that what we're talking about is the neurotransmitter serotonin, and let's say that this is in the brain of somebody who is depressed, right? So one of the things about depression and mood generally is that lower mood and, specifically depression, is associated with lower amounts of serotonin. Now, it's not quite that simple, but it is a big, a really strong association for many people. There's low levels of serotonin, and if you increase the amount of serotonin, it can help with the symptoms of depression. So let's see, how does the drug Prozac do that? Because that's actually exactly what it does. So Prozac is a very common antidepressant, and it is part of a class of antidepressants called SSRIs. And SSRI stands for selective serotonin reuptake inhibitor. That's a mouthful, but you actually know enough right now to figure out what that means. So reuptake inhibitor kind of gives it away. The thing is that we talked about these reuptake mechanisms. This receptor on the presynaptic cell that pulls in that extra neurotransmitter. So if you inhibit that process, right? If you take an SSRI that inhibits that process, what you're going to do is leave more of that neurotransmitter in the synapse. So that is what we're seeing here. So this little red molecule, this little red jagged thing, is a representation of an SSRI molecule, of a molecule of Prozac, basically. And so again, we said selective serotonin reuptake inhibitor. So what it's going to do is travel to this reuptake pump, this mechanism that pulls in extra serotonin, this receptor, on the presynaptic cell, bind to it, and basically just stick in there. And what's going to happen is when serotonin, floats over there to be picked up by the serotonin transporter, the reuptake receptor, it is going to be blocked. It can't get in there. That space is simply blocked, and the serotonin just can't get in. So what does that mean? Well, it means there's more serotonin around. So there's more serotonin to bind the serotonin receptors. So what that effectively does is increase the amount of serotonin in the brain. And so we said that one of the associations with depression is that there's a lower level of serotonin. And so this sort of makes sense why increasing the amount of serotonin could help with symptoms of depression. So it's also good to keep in mind that this is not the case for all cases of depression. Not everybody who has major depressive disorder is going to have lower levels of serotonin. And SSRIs don't work for everybody. So that's something to keep in mind. But we'll come back to that in just a second. Okay, so moving on to the other mechanisms by which the brain gets rid of extra neurotransmitter left in the synapse. So what we're seeing in this picture is, again, extra neurotransmitters, those purple blobs sitting in the middle. Those are extra neurotransmitters that were not bound by the postsynaptic cell. And now what we have is this little sort of dark blue shape down at the bottom left of the synapse. And what is that? Well, that is what we would call a neurotransmitter degrading enzyme. And we call it that because it will bind the neurotransmitter and break it into pieces so that it can't be used anymore. So it's non-functional. And we call that process enzymatic breakdown or enzymatic degradation, basically just breaking down the molecule. But so that is another way that we can get rid of these neurotransmitters and regulate the amount of neurotransmitter in the synapse. Okay, so we talked about Prozac and SSRIs, but there are other kinds of antidepressants. One is called a monoamine oxidase inhibitor, or MAOI. This is a class of drugs, and what they are is, what they do is they interact with these degrading enzymes. And so how that works is very similar to the SSRI mechanism, in that what we've got is this monoamine oxidase. So that is the enzyme that would break down neurotransmitters like dopamine, serotonin, and noradrenaline. And those are all what the type of chemical is called a monoamine. And so this monoamine oxidase is an enzyme used to cut them up. Okay, so let's say that what we've got is a monoamine neurotransmitter, either dopamine, serotonin, or noradrenaline, and we've got too much of it in the synapse. So this monoamine oxidase is there to break it up, like we talked about. But if you take an MAOI, a monoamine oxidase inhibitor drug, what it's going to do is, again, fit in that space and block the enzyme from working. And so the neurotransmitter can't get in, and it just stays in the synapse. And again, we effectively increase the amount of that neurotransmitter in the synapse. And so you can kind of see that MAOIs are much less specific than SSRIs. They target more types of neurotransmitters. And so MAOIs were used a lot more in the past for treating depression, but they have a lot more side effects, as you might imagine, and they're often not as effective or targeted. So while they're still used in some cases, MAOIs are not the most common antidepressant drugs. All right. And then that monoamine neurotransmitter can go bind its receptor. Okay, now on to the final mechanism by which the brain regulates the amount of neurotransmitters. And this is called glial cell reuptake or glial cell uptake. Glia are a different type of cell in the brain than neurons. So you may have heard of astrocytes or microglia before, and those are types of glia. They do a lot in the brain, providing structure in the brain, to providing an immune system for the brain, to regulating the amount of nutrients and ions, and one of their functions is to regulate the amount of neurotransmitter in the synapse. So often in a cell, there's not just two neurons that are part of a synapse. There's a third cell, often a glial cell, that's a part of that synapse and actually provides a lot of the structure around the synapse that allows the dendrite and the axon to stay right up next to each other. And scientists will often refer to a tripartite synapse because there's more than just the neuron and the other neuron. There's the glial cell there doing a lot of important stuff. Part of that is regulating ionic concentrations, and part is, like I said, neurotransmitter concentration. So when those extra neurotransmitters are in the space, that glial cell can, very similar to the reuptake on the neuron, grab that neurotransmitter, pull it in, and basically get rid of it, get it out of that synapse. Okay, so that's it. That is synaptic transmission. And again, if you're interested in diving deeper into this, there's plenty of resources out there, some of which are in the description of this video. But anyway, thank you so much for watching. If you like this video, please give it a like and subscribe to this channel. Also hit the bell icon next to the subscribe button so you never miss anything new from Sense of Mind. Before we go, I want to hear from you. What do you want to learn about neuroscience? What interests you? Let me know. I want to keep this conversation going and keep these videos relevant. This Introduction to Neuroscience series that this video is a part of will basically allow you to go from zero knowledge about the brain to a working knowledge of neuroscience and kind of the main findings, or the most important fundamental knowledge that you'll need to understand how the brain works. So let me know what you think. But we're also coming out with other videos that are less technical and are more focused on giving you the tools to kind of increase your well-being and let you kind of take charge of your life a little more. So once again, thank you so much for watching. Also, don't forget to follow Sense of Mind on social media on both Instagram and Facebook, and sign up for the newsletter. We'll get that going soon. The links for all three of those are in the description below. This video was brought to you by the Diamond Mind Foundation. It was written and produced by me, Andrew Cooper Sansone. Thank you so much for watching. I'll catch you next time.